Hi, I'm Kelsey, one of your grad students for Neuroeconomics, and today I'm going to tell you about the MRI, which we're currently doing a scan on right now. So what better time than to talk about it?
So, have you ever wanted to know what part of the brain encodes happiness, depression, the color blue? That's too bad because we can't use the scanner to say where any of those things are. However, we can ask very specific questions about economic tasks that we understand really well. So, what can we ask? We can use very specific tasks or games, like the ones that we've been studying in neuroeconomics class, to see what is changing in the brain under those certain conditions. But how do we do that and what does it mean? So the MRI is a giant machine that is currently behind me and making noises that emits multiple magnetic fields using these giant coils. That means that whenever it emits the magnetic fields, it excites the protons that are in hydrogen atoms. Whenever it excites them, I'm sorry to trigger anyone that has taken chemistry, it moves the electrons up into the next highest electron shell or electron cloud, whatever you want to call it. Electrons don't like being in that state, so whenever they're up there, after being stimulated, they give off a photon and move down to their normal, nice little electron shell. And the photon is what the MRI can capture to give us nice structural scans like this. Takes about 5 minutes to get this kind of structural image. So we get a three-dimensional image of what someone's brain looks like. However, this doesn't tell us any information about what changes in brain activity are happening. So we need to do a different type of scan to get that information. So the next type of scan we do is a functional scan. This type of scan has the participant perform a task or watch a stimulus on the MRI using a projector and a mirror and react using a button box they hold in their hand. This is the scanner. I can't go in because you're a magnetic device, but that's it. And you can see the little head coil. If I zoom in, there's the head coil, and that's the little mirror that goes on top. And then the projector projects to the back where you see that little fixation cross. So the person goes on this little gurney thing, gets loaded in. They go in there, they can use the mirror to see that little screen that's behind them on the scanner. And that's how it works. We can then use the bold response to then tell what information is happening in their brain. Sorry, what activity is happening in their brain during different parts of the task. So what is the bold response? Basically, the brain needs glucose to do stuff, and it only really keeps a little bit of glucose in the brain at a time. You may remember from biology class that in order for glucose to get used, a cell has to do cellular respiration, and that requires oxygen. This means that when a part of the brain is activated a lot, it uses a lot more glucose than normal, and therefore requires more oxygen, which requires more blood flow. The BOLD response stands for blood oxygen level dependent response. When a part of the brain is just vibing, resting, whatever, there's a set amount of blood flow that occurs. But whenever it's activated, more blood flow goes to that part of the brain. And that's where we get our BOLD response. You'll also remember that blood cells carry oxygen using a protein called hemoglobin. When hemoglobin has oxygen bound to it, it's super magnetic. But when hemoglobin releases its oxygen to the cells that need it for cellular respiration, it's basically not magnetic. This means that when a part of the brain is at rest, it has lower magnetic properties. When it's activated, it's more magnetic. You'll also recall that the MRI is a giant magnet, so it can pick up the presence of magnetic compounds. So we use it to pick up the BOLD response in the brain, those fluctuations in blood flow in the brain. We pick up the magnetic response across the brain in slices, and it takes about 1 to 2 seconds to get the whole brain's 3 dimensions of activation. We call the units of activation voxels. We can decide how long we want it to take to get the entire brain, but longer slices means more spatial resolution, but less certainty in temporal resolution. So we can tell really well where things are happening in the brain if slices are longer, but we're not necessarily as sure what time things are happening at. So you have to kind of have a trade-off. My studies usually have a TR or a magnet response of about 2 seconds. So they're a little bit longer. But it isn't quite so simple.
It takes about 5 seconds to get a bold response due to a stimulus. So when we get our raw data, we need to do some math to make sure our bold response is timed approximately when the stimulus occurred. So you see here, you have an initial dip, and then you get a peak at about 5 seconds, and then the peak kind of goes, it decreases over time. Although if you have an extended stimulus, then the peak will plateau and you'll get an extended point in time, which we have to use in our analysis later. The other thing is that we don't get all of our slices on the brain at the same time. So we have to correct for these slices that occurred at slightly different times in slightly different places in the brain to make sure that they're all lined up so that the stimulus is at the exact same time. We also have to correct for if the person moves their head left or right, up or down, because if we got too much movement, then we'll be looking at a place in the brain that isn't actually there. So if we were going to put all of these steps together, you take your slice timing correction and do that. Then you add your motion correction. You have to do some quick math that I'm not going to talk about. Then you have to co-register your functional scan to your structural scan, so you know where in the brain things are happening. Then you have to normalize it and make sure everything's lined up. And then you also have to align it to a template so that you can tell kind of where in the brain things are. The template that we like to use is the MNI, which is the Montreal Neurological Institute. And that's what we use. And then you also do like some temporal and spatial filtering, but we're not going to talk about that because I don't think you care. Then there's the fun part, which is where you analyze the data. So the first thing you need to do is make a design matrix of when in your task all of the things happened. So in my task, the multi-round trust task, I have when the people start just making a decision about what to invest. I have when people have to make that actual decision, when people are waiting for the trustee to respond, when they get the response of the trustee, and then when they see how much money they made and how much the trustee made. Those are all different times, which could have different things happening in the brain. So once you have your design matrix, you run some statistical models in the brain and look at an individual person's brain, like what changes happened at different times. In the first level analysis, you're aligning the pre-processed data to the design matrix to compare. And then in the second level analysis, you're comparing across people. So you might have a bunch of different people playing the trust task for a period of time, and you can look at what all of their brains did whenever they saw a trustee response. So in our example, the question we might want to ask is, what parts of the brain are more activated when the investor sees what the trustee returned? And so we would pull up a statistical map of that time point and put it in XJView to see differences in activation. So we're going to do that now. Whenever the investor sees what the trustee did. So that's what that part is. So we're going to open up that map. And look at that. We have a nice little brain. You see all this activation going on. So you can click around here to get like different parts of the brain, right? Currently our cluster size is kind of small and our p-value is also pretty big. Now we can look at different areas of activation in the brain. So what areas do we see activated? Looks like this part and this part are activated. We got some up there. We got some of this. One thing I want to remind you of while we're looking at this is that this is the bold response. This is not technically directly measuring electrical activity. We're measuring blood oxygenation levels. So some people take that really personally, and therefore I feel like I have to mention it, but this is the left insula. Do we see activation? Yes, we do. We have activation in both the left and right insula. So whenever they're seeing what the trustee does, just in general, this doesn't ask any questions about, what the return was. It just asks, when they're returning, what's the activation like? We have activation in the insula.
as compared to the other parts of the task. So what does that mean? If the insula is activated, maybe we're getting a negative response. It's not super activated, but maybe if we look at places where there was poor return, this person has negative response emotionally to having bad return. I don't know. So this kind of gives us an idea of what other questions we could ask of our data using more complicated statistical models and more complicated comparisons. So to summarize, that's the scanner, bold response, and how to do imaging analysis.